Hello everyone. Today we are going to discuss the topic 4.1, Importance and Role of Bioinformatics in Life Sciences. So we have to discuss the applications of bioinformatics in life sciences and importance of it. There are the several applications or importance of bioinformatics in life science. Among them, the first one is a sequence analysis. The geneticists, molecular biologists analyze the genome sequence information to understand the disease process. Then second one is a molecular modeling. That is the crystallographers, biochemists design the drugs by using the computer aided tools. Then phylogeny or evolution. For the prediction of the phylogeny or evolution, it is also used. Geneticists obtain information about the evolution of organisms by looking for similarities and dissimilarities in the gene sequences. Ecology and population studies that bioinformatics is used to handle large amount of data obtained in the population studies. Then main medical informatics, then for the personalized medicine, preventive medicine, then in the development of the drug, in the crop improvement, forensic analysis, insect resistance, development of drought and the resistance variety, veterinary science, then climate change studies, crop improvement, and antibiotic resistance organisms. All these are the applications or the uses of the what that is the bioinformatics in the life sciences. These are the what the ways or the what that is the various areas in the biological sciences or in the life science by which where we are using the bioinformatics or the bioinformatics tool. That is the what the DNA sequence analysis by the what that is this is the search engine that is a blast for the what that is a DNA sequence analysis. After the query, it matches the or the, that is the shows the similarities and the dissimilarities. Promoter analysis, then cluster X, gene prediction for the regulatory elements, intron and exon finding, primer designing, codon usage optimization, and virtual translation. All these are the uses related with the what the DNA sequence analysis. Then molecular dynamics simulations. It is that in that the protein DNA simulation, the protein ligand simulation, and the drug DNA that is the simulation medium. Then in drug designing also the bioinformatics plays the key role by the target identification, by target validation, lead identification, lead optimization, admit prediction. Then in bioinformatics, the what that is the sequence analysis or the as the what we are doing the what that is the DNA sequence analysis. Another thing or the important most thing that is the protein sequence analysis, molecular mass, then instability index, then amino acid composition, then identification of the domains, motif search, then signal peptide identification, secondary structure analysis, tertiary and the quaternary structure of the proteins that can be identified with the help of the what? The bioinformatics. Then bioinformatics plays very pivot role or key role in the phylogenetic analysis or evolutionary tracking. The reconstruction of evolution history, tracking the gene flow, identification of the conserved region that is in the phylogenetic analysis. Then in molecular interaction, that is the protein-protein docking, then finding inhibitor activators of protein, protein DNA interactions, transcriptional factors identification, and the protein ligand interaction that occur under the what molecular interaction. And the molecular modeling, that is the 3D structure prediction and the protein function prediction can be done with the help of the molecular modeling. All these are the what the applications or the uses of bioinformatics tools in various areas of the biological sciences. This application it involves the first application among that is the literature retrieval. That for the literature retrieval, the PubMed is the search engine, or the use the PubMed to search the what that is the journals and other literature on any biological or chemical atom of interest. Full articles are not provided in the PubMed in this database. Only citations and abstracts are available to view. So that is the what the literature retrieval can be done with the help of the PubMed by using the what that is a NCBI. Then that is the what NCBI National Center for Biotechnology Information. By using the what that is a NCBI, that you can retrieve the what that is a literature from the what that is a PubMed. Then the biological application, the biological data is stored consistently and is easily available to scientific community. The requirement is then to provide methods for extracting the meaningful information from the mass of data. Among them, the first one is what the nucleotide application information retrieval. That nucleotide application, that is the what the DNA sequencing, or that is the there are numerous databases around the world containing information useful for computational biologists. The main ones are the what the NCBI, that is the what National Center for Biotechnology Information. Then the what there is are the what the databases which are related with the what that is a nucleotide or the sequence based that is the DNA sequence databases, that are the what that is a EMBL, that is a European Bioinformatics Institute Laboratory, uh, European Molecular EBI, that is a European Bioinformatics Institute. Then the DNA database of the what that is a Japan, or the what that is a DDBJ. All these are the what that is the nucleotide applications. These are related with this. Then next to this, that is what the sequence retrieval. The sequence retrieval is what the find the nucleotide sequence for a gene of interest. That is, these are the what you can find the what that is a complete genome by the what that is a sequence retrieval, or the identification of the sequence, or the what that is a nucleotide application information retrieval by using the what that is a the various data banks like the database of the Japan, or the European Bioinformatics Institute, or EMBL, European Molecular Biology Laboratory, or like that. Then sequence identification is done with the help of the what these bioinformatics tools in life sciences to find the function and possible origin of gene from a sequence. The sequence analysis is done, and with this application we can align the two sequences, align multiple sequences, and perform phylogenetic analysis. One reason we would do this to determine what parts of sequence are conserved from one species to the next species. Another reason would be to see how much an organism has diverged from the other organism by simply comparing their DNA sequences. Means two closely related organisms, their DNA sequences can be compared, and you can come to conclusion how much that organism has diverged from the original one. Then sequence translation, that is the what the computational biologists need to analyze their nucleotide sequences, and the best way to do that is to study the protein product. The following programs will either convert your DNA sequence into amino acid sequence, or it will take your protein and convert it into its complementary DNA sequences or cDNA sequences. And these protein and DNA sequences can be analyzed using the other application on this page. Means by using the what that is a bioinformatics, you can what that is analyze the what nucleotide sequence or related with the what that is the proteins, or the what that is the protein products, or that is the sequence which is responsible for particular amino acid sequence. Then the translation, it converts the nucleotide sequences into the what that is a protein sequences. Then back translation, it converts the protein sequences into the what that is a nucleotide sequences or the DNA sequences, or that we called it as a complementary DNA sequences or the cDNA sequences. Then next one is about the protein applications information retrieval. Numerous information retrieval sites on the internet can give very valuable information concerning the sequence and properties of the protein. Numerous databases exist, and each database is accessible through convenient research programs. What are these? The first is a protein sequence retrieval that allows user to retrieve the sequence from protein name, accession number, GI identification number. Then the protein identification that allows user to retrieve a protein name or accession and GI numbers from the polypeptide sequence. So the protein application information retrieval is again one of the important that is the what the use in the what that is the life sciences. Then, by using the bioinformatics, the protein analysis can be done. After obtaining the identity or sequence of the protein, there are several valuable tools that allow further analysis of the protein. Information can be obtained concerning the characteristic properties of from the sequence. The another valuable tool is a sequence alignment application that establishes the degree of similarity between two proteins or the multiple proteins. And you can determine the what protein sequence properties by finding the molecular weight, their isoelectric point, then titration curve, hydrophobicity for particular protein. By identifying these properties, we can determine the protein sequence properties. The protein sequence alignment can be done with the help of the what that is these bioinformatics align a single sequence to sequences in a database. Pairwise sequence alignment or the multiple sequence alignment. In pairwise sequence alignment, align the two protein sequences to each other. In multiple sequence alignment, align many sequences against a single sequence. All these can be done with the help of the what that is a bioinformatics single word. You can say that the protein analysis, determining the protein sequence properties, protein sequence alignment, that is a pairwise sequence alignment, multiple sequence alignment can be determined by using the word is bioinformatics in life sciences. Then next, that is the structure analysis can also be done. The structure analysis is done for the mainly the proteins. Several programs have been created that give scientists the ability to look at three-dimensional shapes of proteins and the nucleotide. And therefore, this protein, to know the what that is the functioning of the protein, the what we must know the what the structure of the protein or the three-dimensional shape of the protein. And examining the protein in three dimensions allows a greater understanding of protein functions as well as providing students with a visual understanding that cannot always be conveyed through the word that is a still photographs or descriptions. Rasmol, originally developed by the Roger Sile to use this program, it must first be downloaded onto your computer. And then CN3D and other 3D structure viewers are the softwares or algorithms that can be used for what determining the structure analysis of the protein by using the bioinformatics. Then the next one is what the next tool is what that is the phylogenetic analysis. That is what the analysis of the what that is evolutionary relationship among the group of the related organisms. The phylogenetic analysis are procedures used to reconstruct the evolutionary relationship among a group of related molecules or organisms, to predict certain features of a molecule with unknown function, to track the gene flow, and to determine the genetic relatedness. And these all could be represented on a genealogic tree or tree of life. The underlying principle of phylogeny is to group the living organism according to degree of similarity. Means we are placing the organism according to their Degree of similarity where at the molecular level. Greater the similarity, closer the organism would appear on the tree. The phylogenetic tools are commonly used to test the various evolutionary hypotheses and become indispensable for functional genomics, particularly when the functions of genes are not known. These are the what that is the tools and the what that is a description where you are using the what that is a which are used for the what phylogenetic analysis. First one is a what that is a mega, that is a what molecular evolutionary genetic analysis. These are the what either tools or the softwares or the algorithms which are used to what that is a determine the what the similarities and the dissimilarities among the what that is a similar organisms or the degree of the similarity at the molecular level. That mega, that is the molecular evolutionary genetic analysis, builds phylogenetic trees to study the evolutionary closeness. Then MolPhy, that is its molecular phylogenetic analysis tool based on maximum likelihood method. Like that, all these are the what that is the, just we have discussed the what that is a tree view, or that is a software to view the what that is a phylogeny. Here we have discussed the what that is a tree. And that is the by using the tree you can identify the what that is a that closer the organism that would appear on the what that is a this tree, that is a this tree. It is a what the tree view software to view the phylogenetic trees with the provision of the what that is a changing view. Then JS3, it is an open source library for viewing and editing phylogenetic trees for presentation of improvement. All these are the what that is the popular tools for the phylogenetic analysis. Then next, by using the what bioinformatics, you can identify the sequences or sequence databases are there by using the word that is a bioinformatics. Biological sequence database refers to vast collection of information about biological molecules such as the what the nucleic acid and the proteins and the polymers, each molecule to be identified by a unique key. The stored information is not only important for future use, but it also serves as a tool for primary sequence analysis. And these databases or sequence databases, basically these sequence databases are of two types. One is a what that is a nucleotide sequence databases or the nucleic acid sequence databases, and another one is a protein databases. And by using the bioinformatic tools, we are identifying the what that is a, we are making the what these large sequence databases, and these databases contain a variety of information and therefore classified into the what primary, secondary, or composite databases depending upon the information stored in this. And this each database has become an autonomous presentation of a molecular unit of the life. So sequence databases can be done or the what that is made by using the what that is a bioinformatics tools. Basically or important is the what that is a protein databases and the nucleic acid databases or nucleotide databases, as well as these databases are again divided into primary, secondary, and the composite databases depending on the what that is a information stored in them. That we'll discuss in the again next class. The primary databases, that is the data in the primary databases, is obtained by the experimentation such as Is2 hybrid assay, affinity chromatography, XRD, or NMR approaches, and such as related sequence or structure. The protein databases, that is a Swiss prot is a protein database, UniProt is a protein database, then that is a this PIR is a what that is a protein database, then GenBank, then EMBL, that is a European Molecular Biology Laboratory, and the what that is a data bank of what that is a Japan, that is a DDBJ. These are the three are the what that is a nucleotide databases, and these three, that is the Swiss prot, UniProt, and the PIR, that is the what these are the what these are the what the protein databases, and the protein data bank PDB, that is a what this is also the protein database. These are the examples of the primary database. The secondary database, it can next is the what the secondary database, it contains the information that is derived from the analysis of data stored in the what that is a primary database, like the what that is a conserved sequence, active sites of protein, secondary motifs, and these database it include the scope, then cath, prosite, e-motif, that we'll discuss later on, and the composite databases, that It comprises the information that is derived differently from the primary sources, and it includes the what that is a NRDB, that is a non-redundant databases, and the what which contains the data obtained from the gene bank, PDB, SwissProt, PIR, PRF, all these are likewise the what that is the INSD, that is the International Nucleotide Sequence Databases, is another example of the what that is a composite database, which is collection of nucleic acid sequences from the what EMBL gene bank and the DDGB. The UniProt Universal Sequence Protein Sequence Database, it represents the what that is another example, which is also collation of sequences derived from the various databases, that is a what PIR, PhD, and the SysProt, and the what that is a translated EMBL sequences, that is a TREMBL. Then next to that, that is what the genome sequences databases. The genome sequence databases, or that is the what the gene bank built by NCPI, is a vast collection of genome sequences over what 250,000 species and 250,000 species, and the what sorry, that is the what 250,000 species. The data from the gene can be accessed through the what NCBI integrated retrieval system, that is the entries, while the literature is accessible via the what that is a PubMed. Means by using the word bioinformatics, we can do the what that is a genome sequence analysis for the what various organism, that is for Genopus levis, then Saccharomyces cerevisiae, the Saccharomyces genome, that can be done by using the what that is all the these bioinformatics tool. Then protein sequence databases, that we have by using the bioinformatics, we have prepared the protein sequence databases, and which are these, that is the most significant protein databases. These are the SwissProt, then that is the Swiss Protein Data Bank, then TREMBL, that is what translated sequences of EMBL, that is what European Molecular Biology Laboratory, European Molecular Biology Laboratory, and that contains the what that is a translated sequence. This is the what the nucleotide sequence database, and this nucleotide sequence database, if you are making it the translated sequences, then the translation of the DNA sequences in the EMBL, that are called as a TREMBL, then UniProt, that is a Universal Protein Resource, PIR, that is a Protein Information Resource, and the WWPDB, that is a Worldwide Protein Data Bank. All these are the protein sequence databases that can be derived by using only the what that is a bioinformatics. All these protein databases we are going to discuss in next class. Then protein sequence databases that we have already discussed, that is the PIR, UniProt, then worldwide PDB, that is a protein data bank. These are the what that is a few protein databases, or the these protein databases, that is a UniProt, then Primo Protein Data Bank, ProSite, then SwissProt, Interpro. All these are the what the databases of the protein databases. Then predicting the protein structure and function can be also done by using the bioinformatics. The protein molecules begin their life as a shapeless amino acid strings, which ultimately fold up into the what that is a two-dimensional and the three-dimensional structure to become a biological activity. Unless and until we know the three-dimensional structure of the protein, we cannot predict the what that is a function of the protein. And this can be predicted, or the three-dimensional structure of the protein, it can be predicted by using the what that is a bioinformatics. Usually the 3D structures are determined by X-ray crystallography or related techniques like the what that is a NMR. The protein 3D structure can be predicted using various bioinformatic tools like the what that is a comparative modeling. Then for the drug designing, also the bioinformatics plays very important role. The many what that is the programs or the software tools or the algorithms are designed for the what development of the drug, and this is a what non-invasive technique, uh, and the what this drug discovery process by which new drug molecules are discovered or designed to cure the different diseases. Bioinformatics has greatly facilitated the complex process of drug designing and playing the vital role in advancing the process of drug discovery because it is faster to analyze molecules on computer as compared to the experimental approaches. And the computer aided drug designing software was developed and has come into existence to discover the novel drug molecule, which is that is a computer aided drug designing. And the whole process of discovering and designing new drug molecule is quite complicated and is quite challenging. The entire process can be divided into four different steps: the identification of the drug target, validation of target, lead identification, and lead optimization. And you can predict the what that is the action of the drug by using the what that is a such type of the what that is a computer aided softwares. So that is many tools are available to predict the drug like properties and admit that is a what absorption, then the distribution, then metabolism, excretion, and the toxicity. These are the five things that we have to observe during the action of the what any drug. That is what the absorption, how it is absorbed, then distribution, then metabolism, excretion, and the toxicity. And for example, the what O series property explorer is a what that is a web-based tool which predicts the a number of admit properties. So this is the very important tool for the bioinformatics is very important for using the war for the what that is a designing the drug. Then molecular dynamic simulations. The biological activities are result of molecular interactions that occur in a time dependent manner. The time-dependent behavior of molecule could be studied using another set of bioinformatic tool, and that is called as a molecular dynamic simulations. What is the role of this molecular dynamic simulation? This aim to provide detailed information on fluctuation, dynamic processes such as ion transport, and small and large scale conformational changes of protein, nucleic acid, and their complex occurring in the biological system. So this molecular dynamic simulation is very important tool or aspect of bioinformatics that plays important role in the world that is a life sciences. This MDS or the molecular dynamic simulation, it can be useful uh, in gaining the insights into situation where use of experimental means is not possible. They also assist in the determination of the experimental approaches like the XRD and the NMR crystallography. Then the few that is what the portals or the what that is the these bioinformatic tools, these are enables the what that is for the drug designing or the what that is a various approaches related with the what that is a clinical applies application. The cancer genome atlas, that is a what TCGA, the data portal. It is a data portal or application. It is a platform and that contains or it is online and it contains a tumor gene expression data together with the clinical information, and by this information it enables the researchers to gather the information on a significant genomic alteration that occur during the development and metastasis of tumor. To help the biomedical researchers to identify gene expression patterns related to breast cancer survival. Means this TCGA that gives us a clear cut idea that is the which gene or the what that is the development of alteration of the which gene results into the metastasis or which results into the that is what the cancer. Then next one is what the predicting the what that is a blood brain barrier, that is a BBB, that is a permeation. It is very much important or essential for drug designing of molecule that act on the what that is a brain or the central nervous system and the spinal cord. Okay, and this the how it is what that is a permeation occurs, and this understanding the process of permeation is very much complicated on the central nervous system, since this compound can cross the this blood brain barrier system by passive diffusion as well as the active transport. As an alternative to invasive animal experimentation, in silico screening methods have been introduced in the development of central nervous system active drugs design and implementation. And this algorithm predicts the what that is a blood brain barrier permeation ability of a given molecule, which provides us an accurate result than the currently available models. So this is one of the software or the software tool that is used for the what that is designing the molecule that act in the what or act on the central nervous system. The next one is what the single nucleotide polymorphism, that we called it as a SNP. That is what single nucleotide polymorphism is the result of variation of single nucleotide at a specific position in a genome. And beside introducing some degree of genetic variation within a population, certain SNPs have been associated with the susceptibility of that individual to the specific disease. And this accumulated knowledge results from the availability of human genome sequences and association of specific SNPs with certain diseases have prompted the development of the predictive preventive personalized medicine. So that is the what the SMP or the what that is a that if you are identifying the what that is a single nucleotide polymorphism, you can what that is a design a drug or the what that is a preventive, you can use that data for the what that is a predictive preventive personalized medicine. So these are the what the three applications or these are the three algorithms that that are of what that is a clinical importance or the what for the personalized medicine, for determining the what that is a drug designing, or for determining the what that is a breast cancer survival, or the how the what that is a which how the what that is a genomic alteration that occurs in the patient of the what that is a breast cancer. So these all are the what that is a important the what the approaches of the what that is a these or uses of the bioinformatics in the life sciences. That is the DNA sequence analysis, molecular dynamics simulations, drug designing, protein sequence analysis, then molecular modeling, molecular interactions, phylogenetic analysis. All these are the what the basic tools of the bioinformatics that are very useful in the what that is a life sciences. Thank you.